Yes, sir. Yes. Sir, namaskar, sir. Sir, namaskar, sir.
Namaste.
Today we have a renowned scientist, professor and chairman, Dr. N.B. Ramachandra, sir. He was chairman board of studies in genetics and genomics, department of studies in genetics and genomics, University of Mysore, Manas Gangotri, Mysore. Sir did BSc during 88 Geology, Mysore University, and MSc Geology, and also PhD in Geology. Thesis title: Contributions to Population, Cytogenetics of Drosophila, Studies on Interracial Hybridization and B chromosomes. So, sir's area of specialization: Drosophila genetics and evolution, human genetic diseases and genomics. Sir has a lot of experience. During 2015, Sir was professor and chairman at Department of Studies in Genetics and Genomics, University of Mysore. During 2017, Sir 7 and professor of Geology Genetics and coordinator for MSc Genetics, Department of Studies in Geology, University of Mysore. And during 1990 to 2007, Sir was a leader at Geology Department. Sir was senior lecturer during 1994. During 2001, Sir was a visiting professor, genetics and development, Department of Zoology, University of Innsbruck, Austria. And also senior research associate during 1999 to 2000 at Cell Molecular and Developmental Biology, University of California, Los Angeles, United States of America. Sir started his career during 1992 as a lecturer at Department of Studies in Geology, University of Mysore, and research associate, postdoctoral fellow at University of Hamilton, Canada, and University of Mysore, and research associate at ICMR Human Genetics, Department of Human Genetics, NIMANS, senior research fellow at SBRR Maharajana College, Mysore, and a research fellow UGC during 1982 at Department of Geology, University of Mysore. Sir also got several awards for his contributions in the field of genetics. Sir was during 2017 elected fellow of Indian Academy of Sciences, Indian Academy of Sciences in Bangalore, and Golden Jubilee Function Award for Science. Sir C.V. Raman Award during 2014. Dr. Raja Ramanna Scientist State Award for the year of his exceptional contribution to science education by Karnataka State Government, and Professor G.K. Manna Memorial Award of ICCA for the year 2014 and 2015. And organizing local secretary for 103 Indian Science Congress at University of Mysore. There was the organizing secretary for the National Congress. And Sir also got award for research publication. Sir C.V. Raman Young Scientist Award, and Sir is visiting professor, senior research associate award, and postdoctoral fellow award at McMaster University, Department of Biochemistry, Canada, Government of India, Nehru Centenary British Fellowship Award from Department of Biological Sciences, Warwick University, Coventry, United Kingdom, and Sir got second rank in MSc, Best Boy of the College Award, Kaveri College, Gonikoppal, University of Mysore. There also completed majorly 16 research projects. And number of PhD candidates successfully completed under Sir supervision 22. Number of PhD candidates currently working 9. And number of MPhil candidates successfully completed 2. And research publications more than 229. Publication in peer-reviewed journals 129. Publication in conference proceedings 9. And gene and DNA sequences in NCBI USA 63. Research notes in Drosophila Information Service USA 14. Books and documentation 14. Presentation in seminars and conferences more than 224.
It's not audible, sir. Hello?
Well, member of KSTA and other my colleagues of KSTA, Dr. M. Ramesh sir, CEO of KSTA, and all the students of today's session. Once again, one welcome you, sir, for giving this talk, sir.
Okay, thank you. Shall I start now?
Yes, sir. Please, sir.
I'm highly delighted to give a talk here and thank all the KSTA members and allowed me to give this talk. Whatever the information is said to make it it's a world information is anyway it's fine, and even the students, if they are really interested, they can write to me to my email is shown here, and also they can visit my website. Okay, so I'm going to share you another 50 minutes about genetics today, where we stand, so on. These are my outline of my talk. Am I audible now, sir?
Audible, sir.
Okay, so there's no issue in my PPT and my the voice.
Yes, sir. Visible and audible, sir. No problem.
Yeah, thank you. So I'm going to review, I'll give an overview of what Mendelism, extension of Mendelism, molecular genetic aspects, human genetics and genomics, where we stand now, and also I'll conclude with this information regarding the achievement and the success stories in the field of genetics. I want all the students to read this, the line, why we look the way we look. Can I get the answer from the student? Anyone? Is there a way for them to answer? Well, this is nothing but the genetics where the genes and the genomes inheriting from generation to generation, from parents to the offspring. So this indicates that genetics is the foundation of all life sciences. I'm going to review in this regard, starting with having the very brief idea about the history of genetics. So genetics can be studied under four subtitle or the phases: Mendelism, molecular genetics, genetic engineering, and the present day genomic era.
Sir, one student, Swati, gave the answer because of the structural genes. He put in the chat box, sir.
Yeah. Okay, yeah, good. Thank you. So I'm not able to see that here. Yes. Oh, in the chat box. Bye.
Yes, sir. Yes, sir. Yes, sir.
Yeah. Okay, good. Yeah. Thank you, Swati.
Oh, welcome.
Yeah. Yeah. Okay. Yeah. What is genetics? Genetics is the discipline of biology, is a science of genes, heredity, and variation in living organism. Genetics deals with the patterns of inheritance from parents to offspring, gene distribution, variation, change in populations, the molecular structure and function of gene, and also gene behavior in the context of a cell or an organism. Last year, we celebrated 200 years of birth centenary celebration of Gregor Johann Mendel, who is the father of modern genetics. And he was born on 20th July 1822. Mendel conducted the experiments using garden pea plants. I hope all the students are aware of the complete biography of the Mendel, how he has studied, how we have got the job and what kind of experiment. Only briefly, I'm going to explain how the Mendel in principle even holds today. So I'll explain them very briefly now. Mendel studied 29,000 pea plants. Out of that, he classified, selected 34 kinds of pea plants. After 2 years, he had 22 purebred lines. Now, out of them, he selected 7 lines of pea which differ from each other with the 7 distinct characteristic features. And he made, by studying this, explaining this, creating the thumb rule of genetics, he was the first person to lay the mathematical foundation of science of genetics. Now it's called as Mendelism. He selected 7 characteristic features of the pea plants, and in all these characteristic contrasting characters, he conducted the experiments, crossing experiment, simple crossing experiment, and he found in the F1 generation, this one of the character was showing the phenotype, and that is called, he has a dominant phenotype, and also he crossed this, inbred them, the characteristic, whatever he got in the F1 generation, he formulated the different laws. I'll go to one after another. The character which has appeared in the F1 generation is called as law of dominance. For example, where in tall and dark plant, we have seen the tall plant, or the yellow or green seed, he could see only yellow seed. All the whatever the progeny he got, all have the yellow seeds like that. All of them, they showed in the second panel is the dominant characters which we have seen in the 7 line of experiments. So he also observed very carefully when any individual produces gametes, the copies of gene separate so that each gamete receive only 1 copy of allele or the factor there, and the other to the other gametes. So every gamete have a single copy of the allele that he called as a law of segregation. When he looked into that, whatever the characters are seen in the F1, they could segregate into 3:1 ratio in the F2 generation. He also taken two contrasting the characters, 2 pairs of contrasting characters, and he crossed them, and he had seen the distribution of traits in the into gametes, and these traits are independently segregated for each other. So he called this a law of independent assortment. I hope all of you are aware of this. The teacher had taught you in your schools and colleges. And the ratio, what we got in F2 is 9:3:3:1. So Mendel's, after conducting, repeating experiments with all these 7 lines, and he could able to establish the principle of genetics. That is, there are 2 possibilities. He could able to disprove the blending inheritance where two characters are mixed and produce the hybrid character wherein the blue and yellow if mix, blue and yellow paint you mix, you get a green paint. That was the one notion had earlier, but Mendel in his experiment and proved that the inheritance is a particulate and discrete factors are inheriting from parents to the offspring. So he disproved the blending inheritance and upheld the discrete patterns of inheritance. So to summarize the Mendel's discovery, laws of inheritance he established. Study approach what he used was experimental and quantitative, and a trace are distinguishing characters that are inherited, and traits are inherited as discrete unit, and he validated them back cross in test classes, and whatever the Mendelian inheritance he conducted, the experiment, it reflects the rules of probability, and therefore Mendel's laid the groundwork for the genetics. When he published his work 1865 in the conference, people have listened, but they are not, none of them are appreciated, not able to understand what Mendel said. That was the time after his presentations of his laws in 1865, 1866 to 1900, the contemporary scientists were discovered the chromosomes and their movements during the cell division, and the contemporary scientists had the better understanding mathematics, and also the accepted wisdom was the inheritance could not be studied in a scientific or statistical manner, where Mendel tried to put forth in a quantitative way the experimental data and probability rules, applying probability rules, but none of the contemporary scientists were not able to appreciate that. So until in 1900, Mendel's laws were not being appreciated or not being practiced. So Mendel died 1884, and he was not happy that because the others are not able to understand or appreciate the work what he has done. So later, three scientists, contemporary scientists in 1900, botanists, independently, that's called Correns from Holland, Hugo Dürus from Germany, and Tushmark from Austria, independently produce the same results of what Mendel did, and also dig out the Mendel's work in the library, and what Mendel already published that work, and they gave the credit to the Mendel, and that has become the rediscovery of Mendelism. And they published that, they given the credit to the Mendel, and this was the big news for the whole scientific community. And William Bateson took a lead, and the UK and the England and the US. He appreciated the Mendel. He became a fan, a follower of the Mendelian contributions, and he put forth, I mean, that was the one that Mendel's idea provided the first comprehensive concept of heredity. Many people who practiced that, they appreciated Mendel, the laws, their experimental data also proving that. And this is the second period. Again, some scientists, researcher, experiment what they have done, they are not able to get that, but however, they got the answer later why it is deviating from the Mendelian, the laws and the principles. So the next stage was how genes are transmitting from parents to offspring during reproduction, how different genes act together to control the variable traits, mechanism of transmission functions of genes were studied, and that has become, the genetics become, the genetics is the most rigorous and conceptualized complete field of the biology, everybody accepted by answering the appreciation of what Mendel experimental experiments and also deviation from the Mendelian principles that were resolved. So, rebirth of the genetics started by the Gregor Mendel, and William Bateson coined the term genetics. He published the Mendel's work, which was published in German text into English, and this was the title. He was published in 1909, and all over the world, people could able to read, appreciate Mendelian experiments. After the 1900 rediscovery of Mendelism, many features, as I told you, the other experiments, some of the experiments were not able to agree with the Mendelian principles, and those are the concepts we call them as a deviation from Mendelian principles, like interaction of genes, multiple alleles, polygenic inheritance, penetrance and expressivity, sex linkage, lethal genes, linkage, and recombinations. These were the not obeying the Mendel's the law, but however, they also contributed understanding about how the genetics is the genetic information passing from parents to the offspring. I'll take some of the example here, one or two examples for each one of them, and I'll try to explain to you. I hope Labrador Retrievers is the dog you have seen, and this dog is the three different colors we find this dog: black, brown, and golden retriever. And all these they are coming from, because these three different colors are produced because of the introduction of genes, black gene and the E gene, B gene and the E gene. If the B gene and E genes are expressed, then completely this dog is becoming black, and only the E gene is expressed, then it's become brown. If the B gene or black gene or both the genes are not expressed, then it becomes the golden retriever. And this tells about the interaction of the genes. The two genes are interacting, produce different phenotypes. I believe you know the human blood grouping ABO system, and I'm not going to detail account of this. However, I wanted to tell that this is the one. Generally, Mendel experiment says that a gene has two alleles, but this is a derivative from the Mendelian principle that a gene, a blood grouping gene, has multiple alleles. There are four different alleles: O, A, B, and AB, and sometimes the AB also can co-dominantly inherit. So there are blood grouping system can become an example for two concepts, like co-dominant inheritance as well as multiple allelism. We use this, and the genes are expressed in all the cell, generally in completely whatever the Mendel tested these traits. But however, here the polydactyly, generally we have 5 fingers, but here is the 6th finger. This is all it's a mutation, and it has penetrations of all the genes to make penetration of this gene to make all the 6 the finger normally. Whereas the other side, the right side, this is expressivity of these genes are not completely. That's why differs in the expression. So this is another deviation from the Mendelian principles. That means all the traits are not expressed completely. Look at the height of these two individuals. These individuals are like a world tallest man, Sultan, and the world smallest man, he Ping. They are 8 feet and 2 feet and 5 inches here. And also we do have variation in the eye color. All this because of polygenic inheritance, polygenic traits. Let us review briefly about the how genetics are taken to the further level, the analysis. After understanding, appreciating Mendel's law and deviation of Mendelian principles, scientists started looking for what is the genetic material, and several classical experiments like Griffith, the MacLeod and Avery, the Hershey and Chase experiments elegantly prove that the DNA is the genetic material, and the DNA, the structure of the DNA, double helix structure DNA, was elucidated in 1953 by Watson and Crick group, and Wilkins and Rosalind also contributed. I believe you know how their DNA structure is arranged by the ATGC and the phosphate and the sugar with the sugar backbone. And later, the scientists were in a race to understand the structure of DNA and the functional properties of DNA, and they are quickly, the biochemist, microbiologist, genetist, all quickly they could able to understand and contribute in many facets and many transactions of the nucleic acid, like the replications, transcription, translation, recombination, mutations, repair, regulations, and the lateral transfer of the DNA from mitochondria to the nuclear genome, from nuclear genome to chloroplast, from chloroplast to the nuclear. All these lateral DNA transfer they could able to elegantly establish in their own organisms, the model systems. That has contributed a lot. 1960s also to understand the structure and the functions of the gene, like the gene is an ultimate unit of function, mutation, and recombinations, and the structure of the gene, the prokaryotic gene, has the regulatory regions and the coding sequences and the stop sequences. It can produce the RNA, and RNA immediately converted into a protein. The simultaneously transcription translation takes place in prokaryote system that we already seen. Somebody already taught you in the college, in your class. Whereas the eukaryotic genes are not like the prokaryotic genes. Eukaryotic genes have little more complex. It has the exons, the coding sequences, and also intervene with the intron sequences, which is non-coding sequences. And these exons and introns are there in the gene. And when the transcription takes place, heterogeneous RNA and with the processing, removing of the introns and the dressing the 5' and as well as 3' and capping and adding poly A tail, mature mRNA have been sent from the nucleus to the cytosol for further translation process. This is the unique way, very systematic and also the complex way eukaryotic genes are arranged. So eukaryotic genes produce different, the length of the protein, the different the isoforms of the proteins, they are formed in different tissues. Like in brain, one of the example of mammalian, the tropomyosin gene has this many isoforms. The striated muscle has the 5' UTR, 3' UTR as a lengthy one, whereas in brain cell, the 3' striated 3' UTR is absent. Even the general 3' UTR also absent there. So different isoforms are formed different tissues. I believe you know there are genes are called in different names, and there are based on their functional properties. Like we do have jumping genes. So I hope you remember Barbara McClintock, where the transposons have been identified in maize and in latent bacteria, and human we call them ALU sequences, and split genes where the eukaryotic genes are having introns and exons. Philip Sharp has discovered this. Edited genes, sometimes the RNA has added later the nucleotide that made the edited gene. Overlapping genes, the same sequences are coded for partially for two genes, one gene as well as some part of the gene is for the another gene, polyprotein gene, nested gene, gene within the gene. It could able to see repeated genes. The rRNA genes were thousands copies are available and they are repeated. Same genes are repeated. Multiple copies are needed. And processed genes, some genes have been made RNA. They put back to the genome itself, and they are called processed gene and non-coding genes. Very, very essential for regulations of the gene and expression of the gene. One of the feature what I wanted, I hope you know that this is the simple pedigree of blood system. Like a blood group man got married to a AB woman, and they produce the three different kinds of the kids. I mean the offspring. The offspring had AB, O, and B. Generally, O blood group is not possible because of this combination of because alleles of every gene has a two allele, and this A, O or A, even if it's not possible to have this. Later, this has been discovered, identified that the O blood group in this couple is possible because the A antigen and B antigens are the one produced in the cytosol, and they have been transported into the membrane, the RBC membrane. It needs another protein to transport them from the cytosol to the membrane. So that is also the precursor H substance, that the H gene is important. If this H gene is absent, even though A antigen, B antigen genes are there, the proteins are produced, they are not able to transport this, the A or the B antigen to the membrane surface of the RBC. That's why they are not able to produce this antigen and show the antigen at the surface of the blood cell. Therefore, the whole blood group in this individual is shown up. That means the precursor H gene is not functional, then the transportation of the A or B antigen to the membrane is not possible. Then we end up with the blood group O because of lack of this gene. So this is generally called as Bombay O blood group. Scientists after in 80s and 90s started studying every gene, the expression of gene and manipulation of gene, functional properties of gene. But in the middle of 90s, they have also identified or discovered the gene profiling method called gene chip, where thousands of genes expression, gene activity, functional functions of the gene can be elucidated. That is called DNA chip or gene chip. This has reduced the working time. The thousands of genes can be screened. This is another technology there. So now we have several gene revolutions. Like we know this 60s and 70s isolation of gene, 60s, 70s, 80s, isolation gene, insertion of genes from one individual to another, manipulation of existing gene, artificial synthesis of gene, transgenic animals and transgenic plants in the 80s and 90s. We have seen that Dolly has been the cloned there, and other many transgenic plants have been cloned individual gene, plants and animals. Genetically modified food, gene therapy, and genome editing. The principles are available, the techniques are available, and this is called gene revolution. So, with the background of this, the advent of the technology as well as understanding genetic principles step by step, from 1900 to the 80s, 1980s and 90s, there are many questions have been asked. And one of the questions, what I'll deviate, slightly deviate to give importance to the human, let us know a little bit about human genetics and the genomics, then I'll conclude that also. I'll come to that point. So we do have different characters, phenotypic characters very easily. You can roll, some of you can roll the tongue, you can see the dimple in your chin, and right-handedness and left-handedness, freckle, and the cliff chins. All these are coded by a single gene. Whatever I said here, this curly hair, all these are recessive genes, the inheritance of recessive gene, whereas attached earlobe is a dominant, this attached earlobe is a dominant gene, it's a recessive gene, whereas the rolling of the tongue, dimple, right-handedness, freckle or curly hair, and cleft are the major dominant genes are involved here. Allergy is again multifactorial, depends on what kind of situation there. So what I'm telling here is that we do see phenotypes of the gene expression here in these cases. So all our traits are controlled by genes, and when you look at our traits, we do inherit traits from our grandparents. We also shared the traits from our parents. We also shared traits with the sibs, brother or sister, and also we do have a unique traits. No one else has that in our family, but we'll have that. That is formed because of a recombination between the gametes, between the parents and the parental gametes, and that's a unique characters we do have in our systems. All these traits are coded by the genes. Any variation, any deviation, any defect, any mutations, if we found in these genes, and that will end up with the diseases, abnormalities. So 4 to 6% of the humanity is with the genetic diseases. So genetic diseases are widely studied. There are 4 different categories: Mendelian disorder, chromosome disorder, multifactorial disorder, non-traditional inheritance disorders. So human chromosome number was the completely known, elucidated only in 1956, wherein 2n equals 46, but long time we have known that as a 2n equals 48. And several different techniques are applied to understand the intricacy of the human chromosomes, dissecting the human chromosome, studying the human chromosome in different cancer patients. So we could able to see how the translocation or inversions or deletions in a different chromosome, that's called sky karyotyping can be done, and with the electron microscopy we could able to see the X and Y, the sex chromosome in the human. So one of the common chromosomal diseases, there are several, about 7,000 genes, single gene diseases have been identified. Some of them are being addressed, treated. One of the intervening problems with the Down syndromes, which has been seen in 1 out of 600 individuals globally, and the Western data tells that the advanced grandmother age is responsible, advanced age, mother age is responsible for this. Non-distinction during gametogenesis is the mechanism behind this. But however, when you look into the Indian situations, the 2 children are born per every hour. Before I complete my talk, 2 children are born with the Down syndrome for majority of the young mothers. And why young mothers are producing more Down syndrome kids? That is the interviewing questions. One of our studies, we could able to did systematically and studied, said not only advanced age mother, even the risk factor is the advanced maternal grandmother age. If it is above 35, chances of producing the the Down syndrome babies, young mothers are more. This is the work what we have done several years ago. And genetic diseases, as I told you, multifactorial, and where the genetic information, the genetic information as well as environment is also responsible for this. When I look at the here, cystic fibrosis is a single gene disorder, is a majority the genetics is involved, whereas the other side, the HIV, the AIDS, is the majority of the environment with the susceptible genes that the genetics contribution, whereas majority of the complex diseases are with the 50% of genetics, 50% environment here. Heart diseases, the psychiatric diseases, mental retardation, all these are with the multifactorial diseases. And now for these, any diseases, the nature and nurture is responsible, not only vice versa here. Both genetics as well environment is responsible to cause the diseases. Now we do have many multifactorial diseases, diabetes, hypertension, and psychiatric diseases. Even our lifestyle is also very, very important here. That will contribute the major environment, trigger our susceptible genes in our genome. So there are ways to control and prevent the prevention programs are available for the genetic diseases. The genetic screening is available here. High-risk genes have been screened. There are several chips or gene chips are available. Premarital screening is available. Prenatal, neonatal, and general population screening is also available. And after screening, genetic counseling is also possible. And appropriate management of and consultation programs also available now to address the issues of the genetic diseases. And now we need more of the increased awareness for the public, the patient, family, and community, and the clinical staffs and health policy makers, and for the public also, awareness of the genetic diseases and their causes and the treatment is very, very essential. That's why I wanted to review this at this juncture. As of now, there are 7,000 genetic diseases are known. Around 5,000 diseases, causative agents are being known, variations, the genes have been known, and about 3,300 disorders are being treated, therapy is found for them, and there are more than 2,000 genetic tests are available now to treat these some of the well-known, well-established diseases, and about 350 the biotechnological products are available to treat these diseases. To review briefly about to address the genetic diseases, 1990, the Human Genome Project has started, and it has been completed within 13 years. They started with America and other countries are involved, having the 5 different objectives: identify all the genes, determine the sequence, the storage, and develop and address the ethical issues. And this has been, working draft has been published in private as well as public funded. The genomic research has been published in Nature as well as in Celera. In Science Journal. The Celera Genomics published in Science Journal, and this has been completed in 2003. The first working draft has been published in 2001, and the complete genome sequences are declared that as April 14th of 2003, this has been completed. The sequencing of this 20 individual heterogeneous genomes has started many questions, and many post-genomic research has started. With this, the genomic research, we have a chromosome by using different staining techniques. Cytological map for every human chromosome has been established. The G band, C band, and the spectral banding has been established here. And using the genetics principles of linkage analysis, recombination frequency, genetic map also been established by log score, the term called using log score, and also physical mapping of the genome sequences by using variety of techniques, restriction mapping, and the artificial chromosome quantic mapping, and STS mapping, and different types of physical mapping are also responsible to map correctly the position of the gene on the chromosome region side. So we do have now the cytological map, genetic map, and physical map where exactly the genes have been located. So I want the students to go, or I want you to take OMIM, online Mendelian inheritance of man. That is OMIM. If you do the Google search, you can find out the genetic diseases and which chromosome, what kind of diseases are there, location. So it's easily available in the NCBI databases. Genomics is the study of the genes and using the excellent methods, isolation of the every chromosome, and completely know the sequence of the complete chromosome and the whole genome. And after the whole genome sequencing, we could able to classified what kind of genetic components are there in our genome. Only about 2% of the coding sequences, about 26% is the intron sequences, and about 20% of the line, the transposons, and 13% of the short sequences, and many of the repeated DNA sequences are there in our genome. This has been classified. That means the human genome is only 2% of coding. Majority human genome is repetitive DNA sequences. So we have 3.2 billion base pairs coding for 25,000 genes, and 99.6% we are identical each other. That means we are different, but 4 million nucleotides. And the genes have been genes, number of genes, and the size of every chromosome has been studied. The first chromosome, longest chromosome, had around 3,000 genes and 279 million base pairs. Look at the chromosome 8 and 8 and 11. 8th chromosome is a bigger, longer chromosome compared to the 11th chromosome. However, the 11th chromosome has almost the double the size of the gene. That means the number of genes and the size of the genome is not going together there. And similarly, here look at the 18th chromosome having 766 genes, and chromosome 19th have double the gene of that, and however the size is less, as most genes are harbored in the 19th chromosome. The gene size varies from 21 nucleotide, that's a microRNA, to longest genes so far known is the dystrophin gene is 2400 kb. The dystrophin which causes the muscular dystrophy in our system. Some of these specific genes and the protein genes coding for a proteins have been identified for uniquely for the human. So after the Human Genome Project, several projects, the post-genomic projects have been launched to understand more about the genomic, because the technology available and the individual genome project, 1000 genome project, ENCODE project, synthetic genomics, personalized genomics, 1 lakh genome project, several 1 lakh genome project started, genome editing, gut microbiome, and now the pangenomic is giving many meaningful information. The latest one from last 2 months back, the information about the pangenomics after 20 years of human genome sequences completed, and they have completed the sequence. The 700 haplotypes of 250 individuals belong to different genetic background, and they added new sequences for equality sequences, because earlier they were not able to completely sequence the heterochromatic region, and it was possible new variation, new sequences have been added to the genome, and the error rate is 1 error in 2 lakhs bases. Some of the scientific breakthroughs I wanted for the young people to know about this. The color blindness is known, and it's correctable. And 9% of the whole humanity is having the color blindness, around 9%. So it's an X-linked recessive gene wherein the red and green color not able to distinguish. For example, in the signal light, we are not able to distinguish red signal, green signal, or red signal. That will end up like gray, and they will not able to distinguish. Now corrective glasses are available. We can save our life using the corrective glasses here. And happiness genes have been identified, the gene which regulate the serotonin. Many of the time, many of the persons, even though have everything, they are not satisfied. Always they cranky, they will not say satisfaction. So maybe they have a defect in this, the serotonin regulation. So such people, depression people, have been treated with giving additional dose of serotonin. They become near to the normal day. I hope you know the Angelina effect. Angelina is a model. She is a model and actor, and she had her mother and aunt had breast cancer and cervical cancer. They died because of the cancer. She has sequenced her own genome, that she found that the BRCA1, the mutations in her genome, and she's made the mastectomy, and she's taking monoclonal antibody, and she is now normal because genomic medicine she's taking now. We do have the sports gene called ACTN3, and which help for regulating the fast muscles, and this can be tested, and we can accordingly we can put the sports person to go for the sports or not. They are fit for sports or not. We do what we are, what you are, what you eat. So now our genes for sensory pathways have been identified. Several genes have been identified for like taste, smell, and texture, and that will be regulated. Give the reward response to the brain, and this has been identified. And the genes that influence what we eat are being identified. This will help to improve the personalized nutritions and to boost the health and prevent diseases, prevent the disease. This is called nutrigenomics, the field. So now with the present day knowledge about the genomics, genomic medicine, and we are getting ahead for the screening of the, not the trial method of the treatment, the screening, predisposition, and preventive measures for any kind of diseases is being taken up. This will be the next order in the near future of the treatment of any diseases. To conclude my talk, true legacy of Gregor Mendel, we have to remember this has been reviewed in Nature in editorial in July 2022, where in his conclusion his paper, he quote this, I quote this: the validity of the sets of laws suggested for Pisum requires additional confirmation. Thus, a repetition of at least the more important experiments would be desirable. So what Mendel did experiment, he repeated in 7 lines. He advocated the other scientists to repeat in other contemporary model experimental organisms. So, and that's why Mendel's have been called as a careful scientist, cautious, patient, and committed to data, allowed him to make the discoveries that have stood the test of time even today. I want all the students to relax and read this. So far, whatever I said, explained about the genetics and is Rajan, that it is coming from our parents, grandparents, and it is going for our great grandkids. I hope you can read, some of you can read this. Ninnajja muttajja ಮೂಲಜ್ಜರಲ್ ರೆಲ್ಲೊರಂ ನಿನ್ನು ಲವತರಿಸಿ ಮುಂದಿನ್ನು ಜನಿಸಲಯ್ಯ ನಿನ್ನ ಮಗ ಮೊಮ್ಮ ಮರಿ ಮೊಮ್ಮಡರು ಜೀವಿಪರ್ ಅನ್ವಯ ಚಿರಂಜೀವಿ ಮಂಕುತಿಮ್ಮ. This has been written by DVG, and even the common man 1943 could able to understand, they appreciate the concept of the genetics, the inheritance, the patterns of inheritance, and this is even holds today. Now our Anvaya Chiranjeevi is the DNA goes from parents to the offspring, generation after generations. So use of genetics today for personalized medicines, the tailored disease prevention and treatment, to breed drought and disease resistance crops, to improve the health of agricultural crops and animals. And globally, more than 25 companies are offering direct to consumer genetic testing services to public. And we are in the verge of several millions of individual sequencing, genome sequencing. We are in the verge of developing the identity cards for our genome characteristics, like lipid profile. We are going to have very soon in the pathology labs or the diagnostic lab, the genome testing or characteristic, the identity card or profiling, genome profiling. So the impact of the genetics, invisible gene was identified, dissected, manipulated, synthesized, edited, but it's no longer a dream. Mendel's simple experiments become a tree of knowledge of genetics. Genetics provides an unimaginable wealth for the human welfare and the biosphere, and therefore genetic occupies the center stage in science and the meeting the grounds of all researchers. Therefore, genetics is center here. Even all the physical scientists can also use the concept of genetics, genetic material like physics, radiation genetics, we have a link. Chemists, by biochemical genetics, we have a link. Biochemistry, biochemistry, physiological genetics, embryology and anatomy through development genetics, and with the plant and animal breeding through quantitative genetics, mathematics, statistics. Without that, they cannot understand the population genetics, the evolution, and behavioral genetics, the animal behavior, or cytogenetics through the cytology. So genetics is a wide field, is a broader field. Almost all the field of science can apply the dynamic field or the genetics. The students, we have plenty of opportunity, think in this direction in your future endeavor. Current role of the genetics, where the Nature Review writes that I like this, the quote: genetics have a particular responsibility to engage with the field and public. To what? To build genetic literacy, addressing misconceptions about race, ethnicity, and ancestry, to ensure the responsible use of genetic and genomic information now and in the future. So the responsibility of the genetist to make the literacy and avoid the misconception and to take the better use of the genetic information and genomic information for our welfare. I'm almost done now. So the take-home message for the students is lessons from learned from the genetics is productive collaboration, curiosity-driven, interdisciplinary approach, even multidisciplinary approach. Genetic diseases are preventable, treatable, and manageable. Genetics should be taught, practiced, and propagated as a foundation of all life sciences. Let us uphold the fire of genetics forever and ever. I want the students to read this, the last the slide. The last page, the last sentence, the last word of disease will be scripted by genetics. If we ignore and it is not going to educate them, the obituary for the ignorance. So everything is possible. I remember here the song sung by Dr. Rajkumar, Agadu Yendu Kaikati Kulitare Sagadu Kelasavu Munde. And this is the one I want my young brains to remember, that explore, exploit all the facility, whatever is available now in the globe. So you have the capacity, you have the ability, you can explore. Thank you.
Sir, Nirmala Irigar.
Yeah.
Sir, we know that 99.6% of genes sequences is the same in all. We identify based on phenotype. But in birds like crow, they are also similar like in phenotype. How we identify, sir?
Now, what I told here, 99.6% of the human.
Yes, sir.
You and me, we have 99.6%. 4 million nucleotides are different between you and me. That's why you look like you and I look me like Ramachandra. So this is because of the genomic variations. So we have only 4 nucleotides: ATGC, adenine, guanine, cytosine, and thymine, right? So these are the one present in different combinations, and also the coding for the amino acids that differ there. So your question is about the birds, right?
Yes, sir.
Yeah. Yeah.
Sir, namaskara. Thank you very much for a very, very scholarly talk. Sure, students would have got a lot of take-home message from your talk.
Yeah.
On behalf of KSTA, thank you very much. Sir, are there any more questions? They got it?
Illa, sir, other questions illa.
If they have any questions, I have my email ID there.
Yes, sir. Yes, sir.
You can write to me also, so I can answer them. I can motivate them also.
Yes.
Any kind of information they want.
Any other question? Yeah. They got it?
Illa, sir. Yavudu chat box illa, sir.
Sir, on behalf of KSTA, once again, thank you very much for your scholarly talk. We have with us Professor H.A. Rangnath, sir, our KSTA member, esteemed member of KSTA. On behalf of KSTA, we are very grateful to you, sir. Sir, for your words. Yes, sir.
Chat box sir.
Sure, sir. Sir, Rangnath sir bardirodu. Excellent overview within 1 hour. Hearty congratulations to you.
Thank you, sir. He is my guru. What all the things I could able to say now because of his guidance, his support.
Sir, tavenadru matadbo, sir. Rangnath sir.
It was an excellent.
Thank you, sir, for attending.
Excellent. It was an excellent overview within 1 hour. Congratulations, Ramesh.
Sir, really, sir.
As I suggest to you,
Sir.
you don't make online lectures of this type.
Yeah.
If students are with the speaker, face-to-face interaction would be better.
Correct.
But so, in Mysore itself, you can organize one meeting
sir.
Where he can speak.
Yes, certainly, we plan.
That would be better.
Yes.
Okay, congratulations, Ram.
Thank you, sir. Thank you, sir.
Ramachandra sir, now you made time for this talk, and the talk was excellent, as our Guru Professor H. Rangnath sir has already told. We are very grateful to you, sir. If there are any questions, we'll be forwarding it to you. Please answer them online. Thank you, sir.
Thank you, sir. Thank you.
ವಿದ್ಯಾರ್ಥಿಗಳೇ, ತಮಗೆಲ್ಲರಿಗೂ ಧನ್ಯವಾದಗಳು. ಡಾಕ್ಟರ್ ರಾಮಚಂದ್ರ ಸರ್ ಬಹಳ ಅದ್ಭುತವಾಗಿ ಜೆನೆಟಿಕ್ಸ್ ಟುಡೇ ಬಗ್ಗೆ ನಮ್ಮ ಉಪನ್ಯಾಸ ನೀಡಿದ್ದಾರೆ. ನಮ್ಗೆ ಏನಾದರೂ ಡೌಟ್ಸು ಕ್ಲಾರಿಫಿಕೇಶನ್ ಬೇಕು ಅಂದರೆ ಇಮೇಲ್ ಕಳಿಸಿದ್ರೆ ನಾವು ಸರ್ಗೆ ಫಾರ್ವರ್ಡ್ ಮಾಡಿ ಅವರಿಂದ ಉತ್ತರ ತಗೊಂಡು ನಿಮಗೆ ಕಳಿಸ್ತೀವಿ. ಅದೇ ರೀತಿ ಹೆಚ್ಚು ವಿದ್ಯಾರ್ಥಿಗಳು ಈ ಉಪನ್ಯಾಸ ಮಾಲೆಯಲ್ಲಿ ಭಾಗವಹಿಸಬೇಕು ಅನ್ನೋದು ನಮ್ಮ ಅಭಿಲಾಷೆ. ಮತ್ತೊಂದು ಉಪನ್ಯಾಸಕ್ಕೆ ತಮ್ಮನ್ನೆಲ್ಲ ಆಹ್ವಾನಿಸ್ತಾ ಇವತ್ತು ಈ ಒಂದು ಉಪನ್ಯಾಸ ಅಟೆಂಡ್ ಆಗಿದ್ದಕ್ಕೆ ಧನ್ಯವಾದ ಹೇಳ್ತಾ ಮಾತನ್ನ ಮುಗಿಸ್ತೇವೆ.
ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ನಾಳೆ ಯಾರ್ದಿರೋದು ಸುತ್ತೇಗೌಡ್ರ?
ನಾಳೆ ಪಿ ನಾಗರಾಜ್ ಅವರು ಸರ್, ವೇವ್ ಆಪ್ಟಿಕ್ಸ್ ಮೇಲೆ ಇದೆ ಸರ್.
ನಾಳೆನು ಡಾಕ್ಟರ್ ಪಿ ನಾಗರಾಜ್ ಅವರು ಬಹಳ ಹೆಚ್ಚು ಎಕ್ಸ್ಪೀರಿಯನ್ಸ್ ಇರ್ತಕ್ಕಂಥ ಅದ್ಭುತ ಉಪನ್ಯಾಸ ನೀಡ್ತಕ್ಕಂಥ ಒಬ್ಬ ಉಪನ್ಯಾಸಕರು. ತಾವೆಲ್ಲರೂ ಆ ಉಪನ್ಯಾಸಕ್ಕೂ ಸಹ ಅಟೆಂಡ್ ಆಗಬೇಕು ಅಂತ ನಮ್ಮಲ್ಲಿ ಧನ್ಯವಾದ.
ಥ್ಯಾಂಕ್ ಯು ಸರ್.